Hello, my name is Lisi Skiba, and this semester I have been working in the Angular lab on a project entitled Characterization of Drug Candidates as Lowe Syndrome Therapeutics. Lowe syndrome is a rare X-linked congenital disorder caused by mutations in a gene known as OCRL1. Distinctive characteristics include mental dysfunction, eye cataracts, and renal dysfunction. There is currently no cure for Lowe syndrome, and the disease is unfortunately lethal. The gene where Lowe syndrome occurs, OCRL1, encodes for a lipid phosphatase and localizes in the Golgi. OCRL1 deficiency results in remodeling of the membrane, changes in cell spreading, and defects in formation of the primary cilia. OCRL1 consists of 4 domains: pH, IPPC, ASH, and ROGAP. Mutations for Lowe syndrome have been found to occur on all 4 domains of OCRL1, but the IPPC or catalytic domain has emerged as a hotspot for many of these mutations, more specifically missense mutations. In the amino acid sequence of this domain, there are many known residues that are important for catalytic function. However, about half of these mutations lie outside of these residues. We did test whether or not these mutations had an effect on enzymatic activity and found that the mutation D451G, which occurs on a non-catalytic residue, did not show enzymatic activity. One way to recover enzymatic activity is through the use of drug treatment. To test this idea, we used the drug 4-PBA. This drug was selected as it was already an FDA-approved chemical chaperone. We did see an increase in enzymatic activity after treatment with 4-PBA, and we also saw an improvement in the Golgi complex fragmentation phenotype. The next step was to find other possible drug candidates. 40,000 different compounds were tested from 3 different libraries. This was carried out by overexpressing and purifying OCRL1 in bacterial cells, then incubating with the drug followed by the substrate. A green dye that interacts with the free phosphatase was added, and the absorbance was measured at 620 nanometers. We selected 3 candidate drugs to pursue from our screens, as well as 4 FDA-approved benzene sulfonamides, which was a chemical structure frequently seen among our screen hits. The overall goal of this study was to characterize the effect of selected candidates on Lowe syndrome phenotypes of OCRL1 mutants in cells. The phenotype that we focused on was the Golgi complex fragmentation phenotype. Cells were seeded and transfected with the OCRL1 mutant. The drug was added after an incubation period, followed by staining with a TGN marker for the Golgi complex. Fragmentation of the Golgi complex was quantified by measuring and dividing the area of the Golgi by the total area of the cell. Here, the red line is the median of the D451G vehicle-treated group, and the green line is the median of the wild-type vehicle-treated group. The values provided below the bars demonstrate the extent of rescue. The images above each bar are representative of TGN images for each group. For many of the drugs we tested, there was a statistically significant dose-dependent rescue of the Golgi complex fragmentation phenotype. Overall, this work furthers the potential for Lowe syndrome therapeutics that directly target the mutated enzyme rather than the downstream consequences. This represents an important advancement as there is currently no cure for this lethal disease. Additionally, if the already approved FDA compounds show promising results, they could more easily be implemented into clinical use. For this study, we focused on the rescuing of the Golgi fragmentation phenotype. However, Lowe syndrome cells demonstrate multiple cellular abnormalities. In the future, we plan to test these drug candidates against other Lowe syndrome phenotypes, such as ciliogenesis, which will allow us to identify stronger drug candidates to pursue for Lowe syndrome therapeutics. Working on this project has been a wonderful experience, and I would like to thank Dr. Aguilar, Jennifer Lee, and the Purdue LSAMP program for their continued support. Thank you.